Hello friends and welcome. Today we will discuss about genetics of mental retardation and color blindness, and our main objectives will be to give a brief introduction of mental retardation, to study about the causes of mental retardation, to study in detail about the genetics of mental retardation, to give a brief introduction of color blindness, to study in detail about the genetic basis of color blindness. So, first and foremost, an introduction about the mental retardation. Mental retardation, MR, was defined by the World Health Organization as an intelligent quotient of less than 70 that is accompanied by adaptive limitations in 2 or more key skill areas before the age of 18. Different classifications have been used to partition people with MR. The most frequently accepted classification is based on performances on standardized cognitive tests. There are 4 classes of mentally retarded people. First one, mild. People with this type of MR have IQ of about 50 to 70. Then second one, moderate. People with this type of MR have IQ of about 35 to 49. Third one, severe. People with this type of MR have IQ of about 20 to 34. And last one, profound. People with this type of MR have IQ of about less than 20. According to this type of classification, the level of difficulties and everyday life needs for the social and psychological support of a person affected by MR can be established. Coming to our second objective, that's to study about the causes of mental retardation. MR has heterogeneous environmental and genetic causes acting in various phases of pre, peri and postnatal development. Prenatal causes exert their effects before birth. Examples include genetic and heredity, toxins taken by the pregnant mother, disease and neural tube defects. Genetics and heredity causes include conditions such as Fragile X syndrome and Downer syndrome as well as phenylketonuria. Prenatal toxins include alcohol, tobacco and drug exposure resulting from the behavior of the mother during pregnancy. Diseases and infections such as HIV can devastate an unborn baby. Neural tube disorders such as anencephaly, where most of the child's brain is missing at birth, and spina bifida, that's incomplete closure of the spinal column, are also prenatal causes of mental retardation. Perinatal causes occur during the birthing process. They include birth injuries due to oxygen deprivation, umbilical cord accidents, obstetrical trauma and head trauma. They also include low birth weight. Postnatal causes occur after birth. The environment is a major factor in many of these situations. Child abuse and neglect, environmental toxins and accidents are examples of postnatal causes. An additional reason for being identified as having mental retardation is societal biases, particularly toward diverse students. Now coming to the genetic causes of mental retardation. Current research have been directed to clarify the genetic base of what was accepted as idiopathic MR. The initial reports about the implication of telomeric rearrangements in MR etiology created the necessity of a screening method. The prevalence of this type of modification was established at 5%, being higher in severe mental retardation group, nearly half of all being de novo mutations. The tendency to find a phenotype associated remained unfulfilled partly because of the reduced number of cases and partly because of the heterogeneity of the rearrangements. Deletion of the majority of N chromosomes was implicated in MR, some causing clinical recognizable syndromes. By contrast, interstitial rearrangements are always clinically expressed by MR and cause recognizable phenotypic features. Submicroscopic deletions are responsible for the learning disabilities. MR can be part of a complex cognitive impairment in disorders caused by deregulations in imprinted genes. Coming to our third objective, that's to study in detail about the genetics of mental retardation. A mutation in a single gene should intervene in the biological mechanism of MR. Abnormalities of genes function may be implicated in almost any biological process that the cell conducts. DNA replication, repair and recombination, translation, control of gene expression, membrane structure and membrane transport, energy conservation and cell communication, the cytoskeleton cell division cycle. In an attempt to classify MR genes, two categories were taken into consideration. One including genes implicated in brain development, neurogenesis and neuronal migration, for which MR may be considered to be a secondary symptom due to brain malformation, and another one having those genes responsible for MR conditions without apparent brain abnormalities. The list of identified genes associated with MR is constantly growing. From the approximately 282 human MR genes, 16% reside on chromosome X, whereas its content represents only 3.37 to 4% of all known and predicted genes. The majority of known mutations in X-linked genes are loss of function mutations. X-linked forms of mental retardation are estimated to cause 10 to 20% of all inherited cases of mental retardation. The first gene to be identified was FMR1 that causes Fragile X syndrome and still remains the commonest single gene abnormality to be identified. The future of microarray technology will enhance the diagnostic possibilities of cases suspected of X-linked mental retardation. We will now discuss about the chromosomal abnormalities related to mental retardation. Aberrations in autosomal chromosome number in live-born babies are restricted to aneuploidies. Trisomy 30, that's Patau syndrome, trisomy 18, that's Edwards syndrome, and trisomy 21, that's Downer syndrome. Malformations can be lethal in the first week of life for newborns with Patau or Edwards syndrome. Downer syndrome is the most frequent genetic cause of mental retardation. Results of standardized intelligence test IQ scores in Down syndrome may vary from low normal to severely retarded 85 to 20%, dependent on the degree of cognitive deficits. Children with Down syndrome may present developmental stages similar to other children in the domain of sensor motor, adaptive and social interaction skills, but with a slower rate. In DS individuals, cell density revealed reduced neuron number in late gestation by comparison with early gestation. The same cellular reduction is maintained from the fetal age through newborn period in the hippocampus, cerebellum and neocortex. Autosomal structural anomalies are also associated with mental retardation. Cry-do-chat syndrome is one of the examples of autosomal structural anomalies related to MR. Cry-do-chat syndrome is caused by a deletion of variable size occurring on the short arm of chromosome 5. The first assumption that any breakpoint on chromosome 5 produces a typical phenotype of CDCS was modified by the new study results. In this way, two distinct regions were implicated, one for the cat-like cry in 5p15.3 and the other for dysmorphism, microcephaly and mental retardation in 5p15.2. In 5p15.2 region, two separate subregions were also mapped, one for childhood facial dysmorphism and moderate mental retardation, and the other for adult facial dysmorphism and severe mental retardation. The involvement in brain development and function suggested by the presence of mental retardation may be sustained by the mapping of different genes SEMAF and CTNND2 in this region. CTNND2 gene codes a protein called delta-catenin involved in cell adhesion and movement. SEMAF gene codes a protein called semaphorin F involved in axonal guidance during neural development. The importance for axonal guidance and neuronal precursors migration during cortical development had been proven in mice, suggesting that SEMAF may be responsible for some of the features of CDCS. Sex chromosomal abnormalities are also associated with mental retardation. Two important examples of sex chromosome aneuploids associated with MRR. Number 1, Turner syndrome, 45X0. Typically, a genetic diagnosis of Turner syndrome had been done in adolescence when puberty fails to install. Although initially disregarded, many years after diagnosis, the patients were considered to have mental retardation. Numerous researches showed lower performance IQ than verbal IQ and deficiency in nonverbal visuospatial areas. Differences in appearance, short stature, and delay in the onset of puberty affect their self-esteem. They also may exhibit adjustment problems, anxiety, and social immaturity. A particular phenotype may be present to a significant number of patients with Turner syndrome called X-linked Turner syndrome. Having a X0 cell line, a ring X, and almost every time a 46XX cell line. And the second one, Klinefelters syndrome, XXY. In boys with Klinefelters syndrome, the diagnosis may be established at birth or may be delayed to late childhood or adolescence, since many of clinical features manifest in this period accompanied by continuous behavioral problems and externalizing symptoms. When present, mental retardation is usually mild. Verbal IQ scores measured in KS children are below average while performance IQ is normal. X-linked mental retardation, that's XLMR. X-linked mental retardation, XLMR, was defined as a monogenic intellectual disability affecting mostly males, having a higher prevalence of MR relative to females. The prevalence of XLMR had been estimated in different studies since 1980. Since the X-linked disorders may be transmitted by an unaffected carrier mother, large families with numerous multi-generational individuals with mental retardation facilitated linkage studies and supported the idea of highly heterogeneous pathology. Fragile X syndrome. Fragile X syndrome is the most common form of XLMR, affecting approximately 25% of all families suffering from XLMR. It is caused by the loss of function of gene FMR1, which maps on chromosome XQ27.3, 55 base pair segment of DNA composed by repeats of the nucleotide CGG. CGG repeat is located in the 5' untranslated region of FMR1. Normal people have 6 to 55 copies, carriers have 55 to 230 copies and do not express the fragile site. In female carriers, the fragile X repeats are expanded during meiosis. In affected males, 230 to 1000 repeats are present and they express fragile X mental retardation. Clinical features include long narrow face, large and protruding ears. Patients exhibit also moderate mental retardation, attention deficit, seizures, and coordination problems. Coming to our fourth objective, that's to give a brief introduction of color blindness. Color blindness, also known as color vision deficiency, is the decreased ability to see color or differences in color. Most individuals who are colorblind inherit the trait. Men are more likely to be colorblind because of the way color blindness is inherited. The gene for the trait is located on the X chromosome. Men have one X chromosome and women have two. If a man inherits the gene for the trait, he will have a color vision defect. If a woman inherits a single gene for the trait, she will not, because the normal gene on her other X chromosome will dominate over the defective gene. Women must inherit the defective trait from both parents to be colorblind. The most common cause of color blindness is an inherited fault in the development of one or more of the three sets of color sensing cones in the eye. Color blindness can also result from physical or chemical damage to the eye, optic nerve, or parts of the brain. Coming to our last objective, that's to study in detail about the genetic basis of color blindness. Color vision deficiency or color blindness is caused when the cone cells are unable to distinguish among the different light wavelengths and therefore misfire, causing the brain to misinterpret certain colors. Mutations in the following genes results in defects in color vision: CNGA3, CNGB3, GNAT2, OPN1LW, OPN1MW, and OPN1SW. More specifically, OPN1LW mutations impact cells that are designed to pick up the red end of the light spectrum. OPN1MW mutations impact cells that detect the mid-range colors of the light spectrum, such as yellow and green. OPN1SW mutations impact cells that detect the lower range of the light spectrum, such as indigo and violet. The proteins produced from these genes play essential roles in color vision. The OPN1LW, OPN1MW, and OPN1SW genes provide instructions for making the three opsin pigments in cones. The opsin made from the OPN1LW gene is more sensitive to light in the yellow-orange part of the visible spectrum, and cones with the pigments are called long wavelength sensitive or L cones. The opsin made from the OPN1MW gene is more sensitive to light in the middle of the visible spectrum, and cones with this pigment are called middle wavelength sensitive or M cones. The opsin made from the OPN1SW gene is more sensitive to light in the blue-violet part of the visible spectrum, and cones with this pigment are called short wavelength sensitive or S cones. Genetic changes involving the OPN1LW or OPN1MW gene cause red-green color vision defects. These changes lead to an absence of L or M cones or to the production of abnormal opsin pigments in these cones that affect red-green color vision. Blue-yellow color vision defects results from mutations in the OPN1SW gene. These mutations lead to the premature destruction of S cones or the production of defective S cones. Some problems with color vision are not caused by gene mutations. These non-hereditary conditions are described as acquired color vision deficiencies. They can be caused by other eye disorders such as diseases involving the retina, the nerve that carries visual information from the eye to the brain, or areas of the brain involved in processing visual information. Acquired color vision deficiencies can also be side effects of certain drugs such as chloroquine, or result from exposure to particular chemicals such as organic solvents. Red-green color vision defects and blue cone monochromacy are inherited in X-linked recessive pattern. The OPN1LW and OPN1MW genes are located on the X chromosome, which is one of the two sex chromosomes. In males who have only one X chromosome, one genetic change in each cell is sufficient to cause the condition. Males are affected by X-linked recessive disorders much more frequently than females, because in females who have two X chromosomes, a genetic change would have to occur on both copies of the chromosomes to cause the disorder. A characteristic of X-linked inheritance is that fathers cannot pass X-linked traits to their sons. Blue-yellow color vision defects are inherited in an autosomal dominant pattern, which means one copy of the altered OPN1SW gene in each cell is sufficient to cause the condition. In many cases, an affected person inherits the condition from an affected parent. Dear friends, this brings us to the end of the lecture. Hope you have enjoyed. Have a nice day.